Good afternoon, all students, and welcome to the second session of the second week. And I welcome all of you for the second part of the cell biology portion, okay, which is going to be taken by Dr. Parth Pandya from Navrishna University. And I'm sure it's going to be an enriching session because in the morning you have studied about cell membrane, and we continue forward looking into cell wall and cell organelles. So to officially introduce Dr. Parth Pandya, I would like to request Dr. Kritika Abhyankar, Assistant Professor, Navrishna University, to kindly introduce Dr. Parth Pandya to the students.
Thank you, ma'am. Good afternoon, all. I, Dr. Kritika, welcome you all to this postgraduate e-lecture series, Week 2, Session 2 on cell wall and cell organelles. To introduce Dr. Parth, Dr. Parth has done his bachelor's, master's and PhD from the MSU University of Baroda, and he has cleared GSLET examination. He has a teaching and research experience of more than 8 years, and he has guided over 12 master theses, and he has 3 PhD enrolled students. He is a recipient of many awards. For instance, he has received Young Researcher Award from Indian Institute of Science, Bangalore, and so on. To his honor, he has completed 1 minor project and ongoing 1 major research project sanctioned from DST. He has more than 12 research papers in the international journals and has took a part in various conferences, workshops, and seminars. He also is a member of professional bodies like SRBC and has presented his work in different international conferences. With these few words, I invite Dr. Parth to give a deliberation on the cell wall and cell organelle with importance to CSIR and other competitive examination. Once again, I welcome you, sir.
Yeah, thank you so much, Dr. Kritika. Now let me share the screen. Yeah. Yeah, so are you guys able to see the screen?
Yes, sir.
Yeah.
Yes, sir.
Yes, sir, but would you please hide the notification bar at the bottom of the screen?
Fine.
Okay, thank you so much.
Yeah. Okay, so a very good afternoon to everyone. As I have already been formally introduced by Dr. Kritika and Dr. Darshi. Now, without wasting much time, we'll be moving forward with cell wall and cell organelles. Now, this is a topic which is, yes, you can say it is asked individually in either 2 marks or 4 marks, but in 4 marks, it is combined with other fields. For example, if we take biochemistry, molecular biology, and so on. So we'll be touching all the aspects of molecular biology and biochemistry with respect to the cell wall and cell organelles. Okay. So we'll be discussing the questions also in each topic, and then we'll be going slowly and further how that topic can be more further be enriched. Okay. So let us start with cell wall. We know that cell wall, they are the main outer layer present in bacteria and plants. Okay. So starting off with cell wall of bacteria, we can divide this, or we can classify the bacteria on the basis of a stain called as gram staining, and on the basis of that we are having your gram positive and the gram negative bacteria. So considering gram positive bacteria, peptidoglycan is the major component. That is around 90% of the cell wall is made up of peptidoglycan, and in peptidoglycan we are having your NAG and NAM residues, which is also called as your N-acetyl glucosamine and N-acetylmuramic acid, which is linked to each other with the help of 1,4-beta linkage. Okay, so with this glycan group, we are having the tetrapeptide which has been attached. Okay, so the residues which are conserved over here are the L-alanine, D-glutamine, L-lysine, and D-alanine form. Okay, so this forms a tetrapeptide which is attached to our overall glycan part. And in some cases, we can also find the L-glycine, okay, instead of excess of D-alanine. So this is the structure which has been there to provide the rigidity to gram-positive bacteria. One of the residue which is called as teichoic acid, it has been present, and which maintains the rigidity as well as resistant temperature, as well as beta-lactam antibiotics are being targeted with respect to the teichoic acid. So if we talk about the peptidoglycan synthesis, okay, so peptidoglycan, we know that it has been made in the cytosol, and then it is getting transferred onto the cell membrane, into the cell membrane. And for that, naturally, we need to have certain various carrier molecules. So bactrophenol is one of the carrier molecule which is being governed, and which takes the peptidoglycan residues from the cytosol to the cell wall. So this is how the gram-positive components are being found. Okay, so if you see in the figure, we are having the peptidoglycan layer which is being found just above the cell membrane. Okay, and you can see the peptide which has been linked to the NAM residue, and NAM is linked with the NAG residue. In between, we are having the presence of teichoic acid, okay, and lipoteichoic acids, which are considered to be one of the important functionality with respect to the rigidity which has been provided to the cell wall of the gram-positive bacteria. In case of gram-negative bacteria, it is only 10% of peptidoglycan, okay, and majorly this has been present outside the plasma membrane. Okay, so I hope you guys must have gone through the plasma in detail in the morning. So this gram-negative bacteria, instead of peptidoglycan, lipopolysaccharide or lipopolysaccharides are majorly present in the outer membrane, and this has a type of antigen called as O antigen or O polysaccharide, and this is one of the important feature for immunologists, where they are targeting, they are driving how the O antigen drives the immune response with respect to human body. Okay. Moreover, it is having a core polysaccharide and an endotoxin called as lipid A. Okay. So over here, the peptidoglycan which has been there, that has been attached by Brunn's lipoprotein, which is present in the outer membrane. So if we see a gram-negative bacteria, a typical form of gram-negative bacteria, we are having the outer membrane, okay, where it is having a lipopolysaccharide, having the three parts, the O polysaccharide, the core polysaccharide, and the lipid A portion. And just beneath that, we are having the peptidoglycan, and then the cell membrane comes. Okay, so this is how we can segregate, or we can compare your gram-negative and gram-positive bacteria. Various questions which comes, they directly either comes with respect to the synthesis of peptidoglycan, or with respect to the type of components which have been present in gram-positive and the gram-negative bacteria. For instance, if we say in 2019, it was asked that for bacterial growth, a single cell elongates in size before it divides into two in a process called as binary fission. So during this particular cell growth, the options are the new peptidoglycan synthesis is required, along with the hydrolysis of bond linking the old peptidoglycan chains. The second one says that is new peptidoglycan synthesis is required, but no hydrolysis of old peptidoglycan occurs. Likewise, the third says that is the old peptidoglycan is completely degraded and is replaced with the newly synthesized longer polymer. And the fourth one says newly synthesized peptidoglycan is utilized to deposit a new layer of peptidoglycan in the cell wall. Generally, what happens whenever the synthesis of peptidoglycan occurs, okay, it is always the old which has been already been present, and the new residues are getting joined to it. Okay, so with respect to that, the answer for this is yes, new peptidoglycan synthesis. That is, overall, it is required along the hydrolysis of bond, and that is linking the old peptidoglycan chain. Okay, so yes, any question? Fine. So yes, so this was one of the example I just wanted to give, that this type of questions I'll be posting, and in this I have already said the answer. But in the next questions, we'll be discussing the answer. So I'll be taking a dialogue session from you. Okay, so you'll be answering which particular part and why it is you think that has been that should be the answer. So whenever you are targeting any question, okay, while reading the question, just formulate yourself that among the 4 options which have been there, try to eliminate one by one option. okay, and then select the best possible option according as per your knowledge, which is fitting that particular case. Okay, so select that particular option which is been the most appropriate, and then go forward. So that is the thing that we need to crack while doing either the CSR net questions or gate questions and so on. Okay, so moreover, we are having cell wall in plants also. So in plants, we are having two type of cell wall. We call as the primary cell wall and the secondary cell wall. So primary cell wall, basically, it is totally being made with the help of cellulose, whereas secondary cell wall, it is nothing but it is the deposition of lignin, okay, and which is deposited in the vessels and the fibers. So if we talk about cellulose, Skirt's reagent is one of the reagent which is used to stain cellulose. So why I am focusing on this particular reagents also, because these reagents are also asking 2-mark questions. So to avoid that particular case, we have missed that particular case. It is a very simple one, and that can be easily recalled. So that's why reagents are also being placed with respect to cell organelles and cell wall. So cellulose, we know that biochemically means biochemically, if we say the structure is it is 1,4 beta linked, okay, that is glucan chains, and it is moreover the primary wall contains the matrix polysaccharide, okay, that is pectins, homogalacturon, and remogalacturon, that is 1 and 2, which is alpha 1,4 linked. Then we are having hemicellulosic and xyloglycans, glucomanins, xylins, and mixed linkage glycans, which are around beta 1,4 linked. So that is making up a complex primary cell wall, and that is eventually giving the rigidity to the plants also. Okay. If we talk about the secondary component, okay, secondary component majorly includes the glycoproteins, okay, and that also includes the enzymes. Okay, the structural protein arabinogalactan, which is found in the gums, and also act as a signaling molecule. So over here you can see the structure. We are having the plasma membrane and the primary cell wall, which is being arranged. So this primary cell walls, they are having the soluble proteins inside that barrel-like structures. Okay, they are nothing but the hemicellulose portion, and this hemicellulose together, they are attached to the cellulose microfibrils which have been there. In short, the cellulose and hemicellulose, they are further connected to each other with the help of pectin, and that pectin overall provides a rigidity to the primary cell wall, and that is attached to the middle lamella. So overall, you can say the cell wall of the plants, it is having your middle lamella, primary cell wall, and the plasma membrane. That is the external component apart from cell wall. Okay, so that was about the cell wall in brief. Just to have a recap, we discussed about the bacterial cell wall, okay, and the plant cell wall. So I am taking only the cell wall where the questions are majorly coming, okay. We can go much in detail, but we are restricting ourselves because we are having a lot of organelles to cover, and that comprises most portion in the CSR net from the cell biology portion. Okay, so if we talk about cell organelle classification on the basis of the membranes which have been present, okay, so if they are being classified on the basis of that 3 conditions, okay, where there is no membrane. So examples are the ribosomes. A single membrane has been there, that is vacuole, lysosome, Golgi Apparatus, and Endoplasmic Reticulum, or they are having a double membrane system, which is nothing but the mitochondria and chloroplast. Okay, so we'll start with the mitochondria first, and to initiate that, we'll be going to the endosymbiotic theory. Okay, now endosymbiotic theory, it is a theory where it is a very good theory, evolutionary theory, where it says that how a eukaryotic cell is being evolving, or how it has been evolved. Okay, so over here you can see we are having an ancestor prokaryote. It is having a cytoplasm and a DNA and the plasma membrane inside it. Okay, lining that, and this plasma membrane starts enfolding itself, and the endomembrane system starts. That is the development of the organelles like endoplasmic reticulum, nuclear envelope, and nucleus. They start. So what happens next is once this ancestor prokaryote, it engulfs a aerobic heterotrophic prokaryote, and this aerobic heterotrophic prokaryote is of small size, which is getting engulfed by a bigger size ancestor prokaryote, and that eventually remains as a it attains a part of that particular prokaryote and it behaves like an organelle. So that was being transformed into a much more complex organelle, which we now say a mitochondrion. Okay. Likewise, if the same, let us assume that ancestor prokaryote, it has engulfed a photosynthetic prokaryote. Okay, so this photosynthetic prokaryote remain in the system, remain in the cell, and then it develop into a plastid, and that plastid, which we know that it is nothing but a chloroplast. Okay, so with this we are moving ahead with the mitochondrion. So a mitochondria, you can see it is having a overall structure where it is a double membrane system. Okay, so this double membrane system, it is having an outer membrane and an inner membrane. Okay, and in between that we are having the intermembrane space. Okay, so from going outside to inside, outer intermembrane space and inner membrane, then comes the foldings or septa-like condition, septa-like junctions which have been there. They are nothing but they are called as the cristae, and this cristae, they are attached to the inner membrane with the help of your cristae junctions. Okay, and this cristae, they are having also have the presence of the complexes which we call as your F1, F0 ATPase. Okay, which functions as the production of ATP. So overall, if we see the length of a typical mitochondria, it is 1 to 2 micrometer, and the diameter is around 0.1 to 0.5 micrometer. Out of all the organelles, the mitochondria is considered to be the powerhouse of the cell. Okay, and the name itself suggests that it is providing the energy to the cell. Okay, so this mitochondria can be stained with the help of a stain called as Janus green stain. Okay, so whenever we need to separate how this particular components came into account, how the scientists, they knew that it is having an outer membrane, an inner membrane, an intermembrane space, and a matrix. Okay, so what they did was they intact mitochondria was being isolated. Okay, and they provided a medium where osmolarity was very low, due to which what happens, that is the mitochondria, it swelled up. Okay, and overall it ruptured. As we know, so by altering the osmolarity, okay, the cell will either swell, the cell will either shrink, and depending upon the solution which we are providing that. Okay, so that eventually release the contents, okay, into the space, that is the intermembrane space, and the inner membrane was found to be intact. So overall, this was being subjected to centrifugation. Okay, so this is one of the technique which is called as your density gradient centrifugation technique. Now this density gradient centrifugation, it is based on, we know, the larger the molecule, it will be go down faster. Okay, it will be faster sedimenting compared to your low molecular weight molecules. Okay, so this density gradient centrifugation, it is separate. It was being used to separate the various organelles with respect to whether it called mitochondria, whether we want ribosomes, or whether we want endoplasmic reticulum. So this centrifugation, that leaves the overall, we found that intermembrane space was being found in the sedimentation fraction. Okay, so intermembrane space can be isolated with the help of that. Further, that the supernatant can be exposed, or it can be further analyzed with the help of further undergoing density gradient centrifugation, and the inner membrane matrix and the outer membrane portion can be isolated. Now this isolation over here, it can be used very well to understand whether which part is behaving likewise. Okay, so it's an endomembrane system. We already classified the mitochondria into that. As we know, it is having the outermost as the outer membrane, the innermost layer, that is the inner membrane, and the inner membrane, and the innermost, it is nothing but the crystal layer. So outermost, it is nothing but it encloses all the outer boundary, and it consists of your 50% of lipids. Okay, so one of the neurotransmitters, that is epinephrine oxidation, has been found in the outer membrane, as well as the elongation of fatty acid chains in this. So this outer membranes, they are having the specialized porins, okay, which drives the proton motive force of the mitochondria. Okay, so protons which have been there, that has been ejected out with the help of this particular porins, and the membrane, mitochondrial membrane potential, that is MMP, that is very well maintain. Okay, so this porins also play a major role while to maintain the membrane potential of the cell and membrane potential of the mitochondria. Then the inner membrane, we are having rich in proteins. Okay, they are also having the import proteins of the mitochondria. They are having more protein compared to lipids, so high protein lipid ratio has been found, and they are rich in phospholipid and cardiolipid. Okay, the innermost layer, that is the septa. Okay, they are nothing but the cristae. They are invaginated sheets which have been found. Okay, moreover, majority of the function, which the major function of mitochondria, that is oxidative phosphorylation and ATP production, that occurs in cristae. Okay. Yes, we are also having matrix. Okay, that is the interior part of the mitochondria, and it is a gel-like structure. It is having majorly the proteins, that is of around 500 mg per ml. The intermembrane space between the inner membrane and the outer membrane, we saw that is, it contains a ratio, majorly the proteins and the enzyme that initiate the cell death. That is, you will study in the apoptosis, where the intrinsic and extrinsic factors come into account, and the apoptosis has been covered. Okay. Yes. So coming on to the question. So which of the following is not a function of mitochondria? A, electron transport and associated ATP production. B is fatty acid breakdown. C, non-shivering thermogenesis, glycolysis and associated ATP production. Yes, can I have the answers for this?
Sir, could it be D?
Yeah, what's your name?
Swapnil, sir.
Yeah, Swapnil, exactly, you are right. So
Because
this is not...
glycolysis, pardon, sir. Sorry, sir.
Yeah, go ahead. Go ahead, Swapnil.
Because glycolysis occurs inside cytoplasm.
Yeah.
So that's why it's wrong.
Exactly, you are right. So glycolysis is one of the biochemical pathway which is occurring in the cytosol, and it is not occurring in the mitochondria. So the ATP which has been produced, that is also in the cytosol, and it is not associated with respect to the mitochondria. Yes, it is linked, but the ATP production with respect to glycolysis is in the cytosol. Coming on to the second question, in mitochondria, cristae act as a site for protein synthesis. Come on, it's a 2-mark question which is very well asked in CSIR Net in 2013. So protein synthesis, phosphorylation of flavoproteins, breakdown of macromolecules, and oxidation reduction reaction.
Oxidation reduction reaction.
Oxidation reduction reaction.
Yeah. So why do you think oxidation reduction reactions?
Because that's the site where the breakdown happens and all the major ATP is produced. Therefore, oxidation reduction reaction.
Okay.
Electron transport chain.
Exactly. So yes, the percentage of breakdown of the macromolecules is the correct answer, but to a major extent, it is to the electron transport chain, which is leading to the donation of your electron, and then overall leading to the oxidation reduction of certain complexes in that. So that is the answer. That is, oxidation reduction reaction is the major site which is acting in the mitochondria cristae part. Okay. So in the endosymbiotic theory, as the ancestor prokaryote, it engulfed the a prokaryote which had its own DNA. So mitochondria overall, with chloroplast also, they possess its own DNA, which is also called as mitochondrial DNA. Likewise, chloroplast DNA, it is different. Okay, it is present in the chloroplast. So mitochondrial DNA, it is circular in nature, and it means it encodes for the enzymes and the certain parts of the ribosomes. If we see mitochondrial peptide, that is 13 in humans, they have been encoded by the mitochondrial DNA itself. Okay, and 2 rRNAs, 22 tRNAs, which have been used in protein synthesis, they have been very well encoded by the mitochondrial DNA. So in this, we are having the non-coding DNA, okay, plus we are having the tRNA genes, which are shown in the blue ones, the protein coding genes, and the rRNA genes. So it's a circular DNA, it has its origin of replication, also called as ORI site, from where your initiation of replication starts. We won't be going in that much detail of the replication molecular biology part, but how that particular means the DNA has been arranged, that we need to know. So overall, if we see the mitochondrial DNA, it is around 16,569 base pair in humans. Okay, and the subunits which it is encoding, it has been labeled over here. For example, the NADH dehydrogenase subunit, which has been there, that is complex 1, that is being encoded itself by mitochondria. Likewise, the ATP synthesis subunits which have been there, cytochrome oxidase subunit which are there in complex 3 and complex 4 and in complex 5. So the subunits which have been there, they are also encoded by this particular DNA of mitochondria. Okay, so with this, we come to the function which mitochondria do. So electron transport chain and oxidative phosphorylation is one of the major function which is found in the inner membrane and the crystal structure which has been linked. Okay, citric acid cycle, which is majorly done in the matrix portion, and in various other cell types, it is also doing the uptake and release of calcium ions apart from endoplasmic reticulum, which it does the sequestration of calcium. So this leads to one of the important functions which is called as your oxidative reduction reaction, which is happening. That is your electron transport chain or oxidative phosphorylation, which is occurring in the mitochondria. So we know it is the major complex are attached to the inner mitochondrial membrane, okay, and the crystal space which have been there. So this particular case is linked with the help of the energetics of glycolysis and TCA cycle. So overall, we are having 5 complexes over here, and these 5 complexes, they drive the reaction in such a way that is the acceptance and donation of electron. Eventually, that electron is passed and a single molecule of ATP is been produced with the help of complex 5. Okay, so starting off with the complex 1, it is NADH dehydrogenase. That is where the NADH, it has been converted to NAD+, where the 2 electrons from NADH, it is going to the quinone system, and that quinone system which has been there, that is getting oxidized, and that oxidized cubic quinone further passes the electron system. So the first NADH dehydrogenase, it has again the iron sulfur cluster, due to which it grabs the electron, and with the help of that, it passes on to the electron to the ubiquinone system. So once it passes, what happens over here is there is a proton which has been there, which has been generated. Okay, that proton is eventually passed from the matrix portion to the crystal space which has been there. So it has been assumed that around 2 electron which has been passed. Okay, so 4 protons that has been electron which has been 2 electron which is passed, that eventually leads to the 4 proton generation in the sister crystal space. So once the electron is passed to ubiquinone, likewise the succinate dehydrogenase, which is considered to be complex 2. So this succinate dehydrogenase, with the help of FMN and FAD from the citric acid cycle, it receives the electron, and that electron is again passed on to the ubiquinone. So this ubiquinone, it is acting as a system where the continuous oxidation and reduction of ubiquinone occurs. Okay, and it's a very complex cycle. We are not going in that much detail, but what you have to remember, that is again there is a FES clusters in that, and which accepts the electron, and that electron is passed onto the complex 3, which is nothing but your cytochrome C reductase complex. So once it has been passed, the proton, due to change in the membrane potential, the proton which has been there in the matrix, it goes outside to the crystal space. Again, 4 protons have been passed out. So once the proton has been passed out, again the oxidized state is being where the electron needs to, it is unstable state. So electron from there, it passes to the cytochrome C, and from cytochrome C to cytochrome C oxidase, where it grabs the ultimate electron. So once the electron is being grabbed, again the proton motive force comes into account, and proton is being effluxed out. Okay. So eventually what happens, this cytochrome C oxidase, which is very highly unstable, as it has grabbed the electron, that electron is passed on to the oxygen, and finally the water molecule has been made. So once this has been there, that has been coupled with your complex 5, that is your ATPase, which is eventually leading to the generation of the ATP formation. So yes, this is a very complex process. We have gone through in very fast manner, but yes, what you need to remember over here is that is the 4 complex, the parts of the complex, how the electron is getting generated, and what is the ultimate acceptor. So that type of questions may come with respect to the electron transport chain. Okay, so coming on to the F1, F0 ATPase, it is nothing but F type of ATPase. I hope you guys have studied in the morning, where we are having different types of ATPase, V type, F type, and P type. Okay, so F type of ATPase, which has been there, it is a type of ATPase which is generating the ATP. Okay, so over here we can find that is we are having the F0 subunit and the F1 subunit. So F0 subunit, which has been there, that consists of A, B, and C. Okay, so they are the three subunits which have been found. So A, B, and C out of three, A and B are of single one. Okay, so A and B together they are attached to it is attached as a peduncle side or a stalk side, which is attached to the F1. Whereas C, around 12 subunits of C is been found. So once the proton goes inside, the rotation of this particular C starts. Okay, so eventually this rotation in the below, it gives a force in the upper part, and hence also upper part also moves. So with respect to upper part, there is F0, F1, sorry, it is having the alpha, beta, gamma, delta subunits, where 3 alpha, 3 beta, 1 gamma, 1 epsilon, and 1 delta is being the subunit found in the F1 part. So once this proton moves in this direction, what happens? That is, this subunit moves in an anti-clockwise rotation. Okay, that eventually leads to the movement of the F1, and that F1 also moves. So once this F1, the gamma subunit of the F1 moves, the ADP and PI, which is there in the beta subunit, they slowly and gradually come closer, and eventually what happens, the phosphodiester bond starts forming, and finally you are getting the ATP formation in the beta subunit. So once the beta subunit, once the ATP is in form, it is getting liberated out, and that ATP can be used by the cell. Okay, so overall if we see the cytosol in glycolysis, we are having one glucose molecule that results in 2 pyruvate. Okay, even 2 NADH and 2 ATP molecules have been produced. Okay, so overall, this pyruvate goes into the mitochondria with the help of pyruvate dehydrogenase and citric acid. It goes into the TCA cycle, where it is converted to 2 acetyl-CoA and 2 NADH. Okay, finally, this acetyl-CoA together it forms 6 NADH and 2 FADH2 plus 2 GTP. So if we see a net result of 2 pyruvate entering into the mitochondria, it is nothing but your 8 NADH which has been produced, plus 2 FADH2 and plus 2 GTP molecules. Okay, with respect to that, the questions are also there. That is, how many ATPs are being formed when 2 pyruvate enters into the mitochondria? When one glucose molecule is being hydrolyzed, or it is broken down, and then overall you need to calculate with respect to NADH, FADH, and GTP molecules. So that was about the oxidation of a one glucose molecule. Likewise, we also know fatty acid beta oxidation also occurs in this, where the example of palmitoyl-CoA, which has been there. So one palmitoyl-CoA, when it is broken down, it gives 8 acetyl-CoA and 7 NADH and 7 FADH2 molecules. So this acetyl-CoA further undergoes oxidation, where 24 NADH, 8 FADH2, and 8 GTP is being produced. So overall, you can say one palmitoyl molecule of fatty acid, which is producing 31 NADH plus 15 FADH2 and 8 GTP molecules. Okay. Yes, coming on to the question. The proton motive force during oxidative phosphorylation is generated in mitochondria by exchanging protons for sodium ions, pumping protons out into the intermembrane space, pumping hydroxyl ions into the mitochondria, and hydrolysis of ATP. Yes, it's a 2-mark question.
Sir, B option?
B?
Yeah, why do you feel B option?
Sir, because transfer of the electron through the electron transport chain pumps out the protons in the intermembrane space, that creates the proton motive force during the process.
Exactly, my dear. What's your name?
Sir, Ankur Singh.
Ankur, yeah, you are correct, Ankur. So he explained very thoroughly that whenever the electron transport chain is happening, okay, due to which the protons is being transferred outside into the intermembrane space, and due to which the proton motive force is being generated. Okay. Okay, so various scientists, they have studied this complex individually by inhibiting the complex activity. Okay, so for example, rotenone is one of the inhibitor which is inhibiting the electron transport change of complex 1 specifically. So electron is not being able to pass from complex 1 to ubiquinone system, and rotenone blocks that. Likewise, amityl again blocks complex 1 in the same manner. The site is different, but the action is same. Antimycin A in the complex 3. Okay, so complex 3 which has been there. So in that case, electron is getting inhibited. Likewise, cyanide which is there, it also inhibits the complex 4. Carbon monoxide, it is also inhibiting complex 4. Azide, it is also inhibiting complex 4. So a simple cyanide or simple azide in picomole of concentration can basically stop this complex 4, and eventually a person can die. So that much dangerous or that much toxic is the cyanide and azide. Likewise, 2,4-dinitrophenol, that is, it is blocking the transmembrane proton carrier. That is, it is an uncoupling agent. Pentachlorophenol, again, it's an uncoupling agent which is blocking the transmembrane proton carrier. And oligomycin, that inhibits the ATP synthesis, that is the F0, F1 ATP synthesis. And so ATP is not being formed, and ADP and PI remains in the beta subunit. Okay. So with respect to that, we are having one question where the question says the match the chemical agents that interfere in oxidative phosphorylation process with their perspective mode of action. Okay, so this was overall, it was being asked in CSIR 2017, and it is basically having 4 blockers. Okay, so inhibitors are there and 5 options are there. So in CSR net, this is one of the common thing where you are having the 4 in column 1 and 5 or 6 in column 2. Yes.
Sir, B is the answer.
B is the answer.
Second one.
Yeah, Swapnil, why?
Sir, I don't know about the other options, but I know oligomycin inhibits the FO component, and that's why the name is given on that O. Basically, the name is given FO. That's what I know.
Exactly. So Swapnil is definitely right, but to be more precise and to be more accurate, at least try to match 2 of the answers. Okay, so that will fetch.
D matches with 3. That's why.
D matches with 3. Yeah, exactly.
Yes, the first component is disrupted.
Yeah, complex one.
And
yeah,
it is
exactly.
it consists of FES cluster.
Exactly. So whenever we are answering the questions, okay, just keep in mind this type of questions will be much more, and it will be encompassing both the fields, cell biology, biochemistry, as well as cell biology with molecular biology and so on. So with respect to that, try to apply your knowledge and try to have maximum options in that case, and then go for the right answer. The next one is citric acid cycle. We know that, as we saw, that is pyruvate which has been made through glycolysis. It combines with acetyl-CoA and enters into the TCA cycle or the Krebs cycle. Okay, so this forms a complex a molecule called a citrate. It's a 6-carbon compound. Okay, and the cycle goes on. So with the help of from citrate, it leads to the formation of isocitrate, alpha ketoglutarate, succinyl-CoA, likewise succinate, fumarate, malate, and oxaloacetate. So among all these steps, we are having the isocitrate to alpha ketoglutarate, where the isocitrate dehydrogenase plays a major role. So this isocitrate dehydrogenase is considered to be the rate limiting step. Okay, so this is one of the crucial step which has been there apart from your pyruvate dehydrogenase, from where the pyruvate enters into the TCA cycle. So these 2 enzymes, they play a major role, and whenever we talk about these 2 enzymes, the ratio of NAD plus 2 with respect to NAD matters a lot. Okay, so this ratio eventually says that whether the cycle will go forward or whether the cycle will halt, or whether there is no operation. Okay, so eventually this we are not going into the structural details of this aconitase kaisa hota hai, isocitrate kaisa hota hai, but to have a glimpse of it, this particular thing occurs in the mitochondria, we know that. Okay, and due to which the excessive molecules which are there, the electrons which are there, they are coming into the complex 1, and then the electron transport chain has been formed. Okay, right. So the next important function is the transport of proteins. Okay, so the transport of proteins in the mitochondria, it is being governed with the help of the mechanism of 2 proteins called as TOM and TIN proteins. Okay, so these are basically the proteins which are associated with either the outer membrane part or the inner membrane part. So whenever we talk about the proteins are being made by the ribosomes and in the endoplasmic reticulum. They are packaged in the endoplasmic reticulum. Vesicles are transported to Golgi, we'll see that. And from the Golgi, it has been passed on to the mitochondria. So each and every protein which has been there, they are having the specialized sequence, which is called as your pre-sequence. So this pre-sequence, which is found in the N-terminal portion, so that has been easily been recognized by your TOM protein, which is a translocase of the outer membrane protein. And this TOM protein, it is nothing but an importer which imports the protein inside the matrix part of the mitochondria. So this protein goes inside that, and the signal sequence is being overall cleaved off. So a second signal sequence will be there, which has been identified by your TIM proteins, and that TIM protein will cleave, and whether that protein will be an integral membrane protein or it will be an intermembrane protein, that has been overall been done. So this TIM protein, if it is we are having the TIM22, that eventually cleaves and it keeps the protein in the inner membrane, integral membrane part. Whereas if the TIM22 is TIM23 has been there, so that will eventually pass that protein to the other parts of the mitochondria. Okay, so these proteins which have been there, mitochondrial proteins, they do the reaction with the help of ATP. So this is one of the non-favorable reaction. Delta G over here is positive. Okay, delta G, whenever I'm talking, it's with respect to the biochemical part. So this delta G is positive. To make that particular reaction more favorable, the ATP hydrolysis happens, and once the ATP hydrolysis has been done, that is eventually the reaction has been driven and the protein is being transported across the different parts of the organ. Okay, so that was all about the mitochondria. We are having the second organelle, which is nothing but the chloroplast, which is being made, which is also being found through endosymbiotic theory, and it is a type of plastid. So if we compare a mitochondrion with respect to a chloroplast is a much bigger organelle compared to a mitochondrion. Okay, it is same. It is having a permeable outer membrane and less permeable inner membrane, which helps in transport of proteins. Okay, together this membrane forms an envelope. Okay, as mitochondria, we are having a matrix portion. Likewise, in this case also, we are having a stroma portion. Okay, which is nothing but it has the some of the enzymes in that. Okay, and ATP is basically it is being made in this particular case. Okay. So chloroplast, as mitochondria has its own circular DNA, chloroplast also has its own circular DNA, and that is eventually leading to the generation of its own protein machinery. So in the chloroplast, you can see the green line structures which have been there. Okay, so this green line outer part, it is nothing but it is the thylakoid membrane which has been there. That thylakoid membrane encapsulates thylakoids, and there are stack-like structures which are nothing but they are called as grana. Okay, so this grana are eventually the sites where your majorly the reactions have been driven in this chloroplast. Okay, so this is an electron micrograph. Okay, just to have a visualization what exactly how it looks, how the grana looks, as these are the stack-like structures, and together they nothing but they are forming a thylakoid. So the chloroplast, it is having the around 70,000 to 2 lakh of nucleotide pairs. Okay, so one of the example over here it has been given, that is liver birth chloroplast DNA, that is around 1,21,024 base pair. As in mitochondria, in this case also there are certain genes which are encoded by the chloroplast genome. Okay, that is photosystem 1 genes, photosystem 2, likewise ATP synthase genes, RNA polymerase gene, the NADH dehydrogenase genes. Okay, likewise the repeats of ribosomal RNA and so on. So this chloroplast, they divide by a mechanism called as FTSZ protein. Okay, so this is one of the GTPase which has been found exclusively in prokaryotes. Okay, so जब भी कोई prokaryotes का हम लोग cell division study करते हैं, we always study the FTSZ proteins. So it is one of the cell cycle protein which is governing the cell division in the bacteria. So scientists, they say that this gene has been acquired by the chloroplast from the ancient prokaryotic bacteria, and that is remaining as a same analog and doing the same function. Likewise, we are having the function of non-cyclic photophosphorylation. Okay, So with respect to that, it is nothing but it is also sometimes referred as a Z scheme, okay, where we are having two photosystems, photosystem 1 and 2. So photosystem 2, it is around 680 nanometer, and photosystem 1, it is around 700 nanometer. So the nanometer over here, it says that it gets excited at this particular wavelength. Okay, so in photosystem 2, it is a magnesium cluster which has been there, and that is eventually leading to the excitation of the electron, excitation of the molecule. So this electron carriers, with the help of electrons, as it is there in the electron transport chain, in this case also electrons have been passed from one complex to the other complex. We are not saying a complex over here, but passed from one molecule to the other molecule. So the carriers over here that plays a major roles are the plastoquinones, the cytochrome B6F complex, and the plastocyanins. Okay, so it is a type of non-cyclic photophosphorylation where you can see the hydrolysis of water overall, it governs the release of electron, and that electron which has been there, so that this occurs when your light basically of this particular wavelength 618 nanometer, it falls on this. So hydrolysis of water starts, and then the electron which has been there, that has been passed from the photosystem to the plastoquinone system. The plastoquinone system, it is passing on to the cytochrome B6F complex. Again, it is Porin-like structure which is diverting or which is driving a proton motive force. So electrochemical gradient has been maintained, and hence your ATP is being generated. Okay, so this electron, once it is passed to the plastocyanin, and that plastocyanin eventually it has been passed to photosystem 1. Once the photosystem 1 grabs this particular electron, it gets excited. Once coming down to the normal state, it releases the electron, and that electron is passed to the ferridoxin. So this ferridoxin is the ultimate one where the NADH, NADP reductase and ferridoxin together, it passes the electron and it forms the NADPH. So NADPH is the ultimate formation. So with respect to that, if we see the cyclic photophosphorylation is governed by only one system, okay, whereas for non-cyclic it is governed by two systems. So in cyclic, only and only ATP molecules are produced, and in non-cyclic, both ATP and NADPH both are being produced. In cyclic one, it is a reservoir type of one where the plant is not able to undergo this a continuous process. So the energy is very, very less. So in that case, the cyclic phosphorylation works, and once the ATP is being produced, then and then only the non-cyclic photophosphorylation starts. Okay, so that is one of the mechanism which is operating. Apart from that, we are also having the C3 and C4 mechanism. Okay, coming on to the simplest question. In again a 2-mark question. In chloroplast, the site of coupled oxidation reduction reactions is the A, B, C, D, outer, inner, thylakoid or stomal space. Yes?
Sir, C.
Sure. What's your name, my dear?
Sir, Burhan.
Burhan. Why you are thinking C is the answer?
Sir, because the photophosphorylation cyclic, non-cyclic takes place here, so that's why.
Exactly. So basically, the coupled oxidation reduction reactions, they majorly occur in the thylakoid membrane. Okay. Right. So we are having the C3 Kelvin cycle, C3 cycle. So this molecules which have been there, they are getting accumulated, and it forms a structure called as rubisco. Okay. So this rubisco, which has been there, ribulose 1,5-bisphosphate, that rubisco, it has been transferred, or it has been forming into the first phase of reaction, which is called as carbon fixation. So 6 molecules of 3-phosphoglycerate has been found, glyceraldehyde has been found, where the overall fixation of carbon happens. Okay. In the second step, what is happening, using the energy driven reaction, this reduction of phosphoglyceraldehyde, it goes into glyceraldehyde 3-phosphate, and again 6 molecule remains. So what has been done, that is, once the carbon has been fixed over here, that has been reduced, okay, to more stable form, and then from that again it has been leading to the generation of the RUBP, that is your rubisco 1,5-bisphosphate. So that RUBP, that is ribulose 1,5-bis phosphate is converted to again 3-phosphoglycerate with the help of your Rubisco enzyme. Okay, so overall in the process you can see it's a very energy conserving process. That is, your 6 ATP is being used for doing reduction stage. Again, the NADPH is also been used to do a glyceraldehyde 3-phosphate formation. But once the generation of RUBP, it leads to the for generation of RUBP also the ATP is being used to form the ADP. So overall, you can see the Kelvin cycle, it plays a major role whenever the excessive molecules are there. So for that, for storage purpose, this particular condition has been found. Likewise, we are also having the C4 cycle, which has been found in the mesophyll and the bundle sheet cells. Okay, so mesophyll and the bundle sheet cells, which are there, where in this case, your phosphoenol pyruvate, that plays a major role, and phosphoenol pyruvate carboxylase is the enzyme, rate limiting enzyme, which is the key player in converting the phosphoenol pyruvate to oxaloacetic acid. So once the carbon dioxide which is coming inside and it is converted to your HCO3 minus, so the PEP has been formed and it forms the oxaloacetate, and this has been undergone in the chloroplast with the help of a transporter OMT1. So this oxaloacetate, it is converted to your malate dehydrogenase into the malate, and that malate eventually it goes into the bundle sheet cells. So over here, this malate has two fate on the basis of the ATP and NADPH. If the energy reservoir is frequent enough, it can undergo the Kelvin cycle. If the energy reservoir is less, it will convert again into the pyruvate. The pyruvate is transferred from bundle sheet to the mesophyll, and phosphoenol pyruvate will be produced. Okay, so the commonest questions which occur, which are being given in the CSIR net as well as in GATE and so on. So where does this particular reaction occurs? Whether it's a mesophyll or bundle sheet cell. Likewise, each and every molecule which has been there, pyruvate which is found, it is found in the mesophyll cells or it is found in the bundle sheet cells. Okay, likewise, phosphoenol pyruvate kahan pe milta hai? So that type of similar questions are being very well asked in the 2-mark question in the CSIR net. So if you differentiate between your C3 and the C4 cycle, C3 cycle is found in almost all the plants, whereas C4s are majorly restricted to the monocots, like sorghum and maize. Okay, in this case, the first stable compound, it is a 3-carbon compound, whereas in this case, it is a 4-carbon compound. As we saw, it was PGA in this case, and in this case, it was oxaloacetate. So the primary carbon dioxide receptor in C3 cycle, it is a 5-carbon compound, that is ribulose bisphosphate, whereas in this, it is nothing but a 3-carbon compound, that is phosphoenol pyruvic acid or pyruvate. A single carbon fixation occurs over here in C3 plants, whereas in C4, double carbon fixation occurs. Okay, the carbon fixation is naturally a single molecule ho raha hai, so it is less efficient and slow, whereas in this case it is more efficient and fast because it is just recycling inside the mesophyll and bundle sheet cells. Okay, the fixation of this it requires 3 ATP and 2 NADPH, whereas the fixation of this it requires 5 ATP and 3 NADPH. Okay, it is not being shown over here, but it is requiring this. So granular chloroplasts are involved. In this case, granular and agranular chloroplasts both are involved. Okay, C3 it is only occurring in the mesophyll cells, whereas in C4 it is C4 it is both in mesophyll and bundle sheet cells. The optimum temperature range for C3 to occur is around 20 to 25 degrees Celsius, whereas for this it is 30 to 45 degrees Celsius. Whenever you provide oxygen, C3 is inhibited. No inhibition has been found with respect to your C4, and it can be saturated with the help of sunlight. That is, C3 won't happen properly, whereas if sunlight is not there, it doesn't saturate. It is having no effect. Okay, so coming on to the question, which one of the following reactions takes place during the reduction phase of the Kelvin-Benzel cycle? So as we know, we are having 3 phases. कौन से 3 phases? Which are the 3 phases which we studied?
Carbon fixation,
reduction and regeneration.
Yeah, fixation, reduction, and regeneration. So in this case, we are asked for reduction. So on the basis of reduction phase, just tell me which is the correct answer. Yes?
Second.
Yeah, exactly. It is the second. It is the second one, that is 1,3-bis phosphoglycerate to glyceraldehyde 3-phosphate, where the reduction of this particular molecule is occurring. Okay. Okay, coming on to the second one, the following statements were made with the assumption that the concentration of 3-phosphoglycerate is high inside the chloroplast of an actively photosynthesizing leaf. There will be a high concentration of triose phosphate in the chloroplast. That is the first statement which was given. The activity of ADP glucose phosphorylase will be inhibited. Third, the carbon flow will be diverted from sucrose to starch. Okay. Starch synthesis will be inhibited and carbon flow will be more towards sucrose synthesis. Okay, now we know that it's the starch is one of the storage polysaccharide which is being used in case of plants. Okay, and sucrose is the molecule which is being used either for synthesis or breakdown of sucrose occurs. So now in this particular question, we are having the concentration of this 3-phosphoglycerate which has been there in the chloroplast, and on the basis of that, the formal assumptions have been made, A, B, C, D. So now we have to relate two things. That is, we know that 3-phosphoglycerate has been formed. वो कब form होता है, कैसे form होता है हमने देख लिया. कब form होता है, that we have to now list down. Okay, so that assumptions are that is there will be high concentration of triose phosphate in the chloroplast. Okay, triose phosphate अगर बहुत सारा है, so naturally हो सकता है that it will combine with RuBP and it can lead to 3-phosphoglycerate. That can be the possibility, yes. Secondly, the activity of ADP glucose phosphorylase will be inhibited. Now, ADP glucose phosphorylase, which has been there, generally isme ADP which has been there, it is not found in ATP form nahi karta hai. So that is been driven by kinases. It is not through with the help of this particular enzyme. The carbon flow will be diverted from sucrose to starch. Okay, so ऐसा कोई mechanism नहीं है कि भी phosphoglycerate अगर form हो रहा है, okay, it is found more so that the sucrose से starch पे shift होगा. That is not a mechanism. The fourth one, starch synthesis will be inhibited and carbon flow will be more towards sucrose synthesis. Right. So starch हमने बहुत सारा starch बहुत सारा है अगर cell में, so that needs to be accumulated then to synthesize. Okay, and if the synthesis is inhibited naturally, एक mechanism कम हो रहा है तो दूसरा mechanism ज़्यादा होगा. That is sucrose का synthesis होगा, and due to which that sucrose will be transported from one part to the other part. So in this question, two things are being driven, or two things are being told. Okay, so we have to link the two things, and on the basis of that, the answer is nothing but yes, the triose phosphate it is which has been more, and with respect to that, the starch and sucrose play an important role. Yes, any question till now? Okay, so we are moving to the next organelle, that is an important organelle, that is your endoplasmic reticulum. Okay, this organelle it's a membrane bound organelle which is having the more than half of the total membrane of an animal cell. Okay, so in this we are having the branching tubes and flattened sacs which are extending throughout the cytosol. The tubules are being interconnected with each other, and it forms a continuous row of sheet which is enclosing the internal space. Hence, it is called as your ER lumen or ER cisternal space. That is around it therefore occupies a 10% of total cell volume. So whenever we classify endoplasmic reticulum, we are having rough endoplasmic reticulum and smooth endoplasmic reticulum. So rough endoplasmic reticulum, it is nothing but it is pre-bound ribosome to the cytosolic phase, okay, of this. Whereas in smooth, we are not having any type of bound of the ribosomes. So ribosomes are absent in this case. Rough may we are having protein synthesis that indicates the secretory integral part, means the secretory proteins, integral proteins, and soluble proteins, they are being produced over here. Whereas smooth endoplasmic reticulum, they are being found exclusively for the lipid synthesis. Okay. Yes, rough endoplasmic reticulum, it has been where the maximum protein is being used. Okay, वहाँ पे ज़्यादा मिलेगा. For example, pancreas, salivary gland, जहाँ पे enzyme production ज़्यादा है. So in that organs or in that tissue, it will be more. In smooth endoplasmic reticulum, it is found in liver, adrenal, and skeletal muscle. So smooth endoplasmic reticulum, it is one of the organelle which also does the detoxification type of thing, and that's why in liver it is majorly found. Okay, so if we want to stain ER, a fluorescent stain, that is DIOC, that is dihelioxa carbocyanamide iodide, that is a stain which is being commonly used nowadays for visualizing the endoplasmic reticulum. So as we saw in the mitochondrial fractions, we isolated different fractions of mitochondria in the same endoplasmic reticulum. If we want to isolate different parts, whether it's a smooth one or a rough one, okay, we can do the gradient centrifugation. So in this case, the gradient is being maintained by the addition of a solute called as sucrose. So we homogenize the tissue, okay, and that homogenization makes the rough endoplasmic reticulum and smooth into a particular shape where circular to oval shapes are being found. Okay, so rough and smooth endoplasmic reticulum, they aggregate into small particle-like structures. These particles are called as microsomes, and these microsomes, they can be separated on the basis of the molecular weight, and we provide sucrose in such a way that is a gradient is being created, and that is being further subjected to gradient centrifugation. So due to inertia overall, the larger molecules will definitely, the rough molecules or the rough microsomes which have been there, they will tend to move, they will sediment fast, and they will go to the bottom as they are heavier, that as they are having ribosomes on it. Okay, whereas smooth, they are less denser compared to the rough one, and hence they will remain in the upper portion. So this breakage of the ER fragments which has been there, they are nothing but the closed vesicle-like structures. They are termed as the microsomes, and these microsomes are further termed as on the basis of ribosomes. If they are having ribosomes, they are called as rough microsomes. If the ribosomes are absent, they are called as your smooth microsomes. So in rough microsomes, around more than 20 proteins and more densely packed structures are there, whereas in smooth microsomes, less than 20 proteins are being there. So the another function, the first function of this endoplasmic reticulum is to translocate the protein. Okay, so we know that the protein synthesis happens with the help of ribosomes, and that protein which have been made, that is coming into the endoplasmic reticulum, and that is being translocated to the different parts through Golgi. Okay, so two mechanisms are being found with respect to the translocation. One which is called as co-translation translocation, and the second one is called as post-translation translocation. Okay, so co-translation translocation, it is nothing but it is the mechanism where the ribosome comes, okay, with the mRNA, okay, and with the growing polypeptide chain. So it is getting attached to an endoplasmic reticulum membrane, and वहाँ पे ही आपका translocation starts. That is protein which has been made, that is going inside it. Okay. The post-translation translocation is protein is made outside the ER, attachment nahi hai, and that protein, once it is being made fully, that is getting attached, okay, then it is getting translocated, and once it is getting translocated, the further process starts. Okay, so a simple one, that is what is happening is that many ribosomes are being there. ऐसा नहीं है कि only one ribosome is there and one at a time is coming. So continuous flow of ribosome is there with the mRNA and the amino acid tRNA which is being charged. Okay, so you can see over here the ribosome which has been there, it brings the polypeptide, okay, the nascent polypeptide. So this polypeptide slowly and gradually grows. So it's a type of co-translation translocation mechanism. So this is being grown, and once it is being grown, the peptide is being cleaved off and the ribosome goes away. Likewise, the subunit get dissociated and the flow of this particular thing is being maintained. Okay, so अभी पता कैसे चलता है कि भाई ये प्रोटीन जो है इधर ER में ही लेके आना है. So naturally there is a recognizing particle which is called as the signal recognition molecule, which identifies that this particular signal है तो उसको ER के पास ले जाना है, and after taking it to ER, it eventually starts translocating over there. So once the stop codon is there, stop codon आया वहाँ पे वो signal peptidase will come and it will cleave off the protein, and that mature polypeptide will be translocated in the ER. Then the further mechanism starts. Okay, so in short, if we see the endoplasmic reticulum signal sequence comes, okay, it drives means with the help of ribosome it is driving. The translocator takes, it forms a pore, the translocation of polypeptide has been there. The peptidase comes and it cleaves the polypeptide, cleaving the signal sequence, and the mature polypeptide has been found with the help of in the endoplasmic reticulum. So this particular whole thing which has been there, that is called as a signal hypothesis, which is driving the translocation of protein inside the endoplasmic reticulum. Okay. So as I said, कि भाई signal को कैसे identify करते हैं? So that signal which has been made on the growing polypeptide chain, that has been identified by the SRP. Okay, this SRP which has been there, it identifies the signal. There is a conformation change of this SRP, due to which the conformation change is being identified by the receptor of SRP, called as SRP receptor, and there is a translocon which has been formed. So once the translocon which is formed, eventually the peptide is being attached to that, and the overall peptide goes inside the ER lumen of the endoplasmic reticulum. So once the peptide is going inside, the signal peptidase cleaves off the peptide, and the ribosome subunit is getting dissociate. Okay, and the similar function occurs with the other ribosomes. Okay, so if we see the translocation which is happening, so translocation mechanism, it is found in all the three, that is bacteria, archaea, and eukaryotes. Okay, so we are having a complex which is nothing but a translocon, that is called as a Sex61 complex. Okay, so this Sex61 complex, it forms a translocon which forms a pore-like structure, and that is being identified by, which has been driven by the SRP, जो translocon के पास लेके आता है. Once the pore has been formed, the polypeptide is formed inside, and the polypeptide will go in the ER lumen. Okay. In case of post-translation translocation, what is happening is the eukaryotes which has been there in eukaryotes, the complex which is there, that is 62, 63, 71, and 72. This is the whole complex which has been found. Okay, so simultaneously this complex, it is stabilizing the 61 complex together, and it is forming a larger pore through which the polypeptide is going inside. Okay, so once the polypeptide is going inside, okay, there are certain proteins which are called as chaperones. Now these chaperones will bind to this polypeptide, growing polypeptide, and it will stabilize it. Okay, उसको denature नहीं होने देगा, उसको more folded form में करेगा. Okay, so it will fold the protein, and so that the protein won't get denatured. So this BIP protein are nothing but they are the ATPase or the ATPase chaperones which uses ATP and fold the protein. Okay. In case of bacteria post-translation, the this translocon complex, that is Sex61 complex, there is a homologue likewise which is called as Sec A ATPase and Sec Y complex. So together this Sec A and Sec Y, it is found in the bacteria, whereas in this case it is your Sex61 complex, which includes 71, 72, 63, and 62. Okay, so once the protein has been formed, over here you can see in case of eukaryotes, the chaperone mediated folding is being generated in the ER lumen, whereas in this case it is nothing but in the extracellular, in the cytosol itself, where the ATP is hydrolyzed and the folding happens. Okay, so once the total protein comes inside, okay, the total polypeptide is been coming inside. Once it is getting folded, there is a stop transfer signal. Stop है कि भाई इसके आगे protein नहीं है. So from that, the peptidase will cleave that particular stop signal, and the protein will be the nascent form. So once this protein has been made, there is a modification called as post-translation modification. So this post-translation modification, it has been driven with the help of a molecule called as dolicol. Okay, so this dolicol, it generates the modification via two manners. Okay, so there are two modifications which has been driven. One which is called as N-link oligosaccharide, means the N-link glycosylation, and the second one is called as O-link glycosylation. So N-link glycosylation is occurring in the endoplasmic reticulum, whereas O-link glycosylation, it occurs in the Golgi complex. We'll see in the next few slides. So dolicol, which has been there, it uses the energy with the help of CTP and UDP. So this dolicol, it is nothing but it is behaving like it has the oligosaccharide transferase activity, and this it transfers the OH group from the serine and threonine to the amide group of asparagine, which is the N-link part. Okay, and that is a sequence which has been asparagine X serine and asparagine X threonine that has been identified. So X over here stands for any amino acid which has been there. Once it has been there, that has been the growing your glycan molecules which have been there. So that is being transferred from the cytosol to the lumen. That is called as with the help of flippase enzyme. This flipping happens, okay, and this flipping, once it has been there, that eventually attaches the glycan molecule to the polypeptide. So overall, if you see the N-acetyl glucosamine mannose residue and glucose residue are being arranged to the end part of the asparagine, okay, and that's why it is called as your end-linked glycosylation. Now this is only and only for glycoproteins, okay. So majorly questions which comes जहाँ पे glycoprotein storage disease होता है, so in that case कि कैसे ये post translational modification में defect आता है, that type of questions are being coming, okay. So once the proteins have been made, they are being translocated, they are being folded. Now apart from the big proteins, there are other chaperones which mediate the role. That is the lectins, that includes calnexin, glucose residue, and calreticulin. Likewise, peptidyl proline isomerase, which drives the folding of this particular proteins. So we are having the three molecules which have been there present on the lumen: IRE1, PREK, RK, ATF6. They all act as one of the chaperone type of mediators, okay. So they have the kinase activity, and due to which the phosphorylation on itself, they activate their own self, and the reduction of protein overall it has been forming. So once the protein has been formed, the signals are given by the mRNA for the regulatory proteins like 1, 2, and 3. These proteins which have been there, that increases the activation of these chaperones, which further increases the protein capacity of ER. Okay, so coming on to the question, which of the following statements were true regarding endoplasmic reticulum? It provides structural framework to the cell. It acts as an intracellular transporting system. SER is involved in the synthesis of lipid. All of the above.
All of the above.
All of the above.
Yeah. So all the activities like structural framework of the cell, as well as the intracellular transporting system, as well as the lipid synthesis, is being governed in the endoplasmic reticulum itself. Okay, so as I said, the protein is translocated in the endoplasmic reticulum. Now it needs to pass through the Golgi apparatus. Okay, so this Golgi, which has been there, that is mediated with the help of part called as vesicle. So these vesicles are of three types: COP1, COP2, and Clathrin vesicles. Okay, so COP2 vesicles which are there, they take the protein which has been there, folded protein, from endoplasmic reticulum to the Golgi complex. COP1, it goes, the Golgi से वापस ER में लेके आता है, so retrograde or retrieval system में, and Clathrin coated from the Golgi to the other part of the cell, वहाँ पे ले जाता है. Okay, so now this totally depends upon the type of protein which has been there, and that type of protein which has been there, that has been identified this by the specialized protein which are there in the vesicles. Okay, so वो vesicle में protein identify करेंगे, and then it will pass on to the Golgi and Golgi to the cytoplasmic part. So if we talk about Golgi apparatus, it is also called as Golgi body or Golgi complex. Again, it's a membrane bound organelle, and flattened stacked pouches called as cisternae has been found. It is responsible for various transport mechanism, modifying mechanism, likewise O type of glycosylation which we talked about. Then lipid delivery system for targeted destinations. Okay. Then it is just located to the endoplasmic reticulum and near to the cell nucleus. Okay. If we talk about in plant cells, so carbohydrate synthesis may hemicellulose and pectin which have been made, it is totally in the Golgi complex. So fluorescent ceramide, which has been commonly used in the Golgi for staining with respect to plant and animal cell. So coming back to what I said, COP1 and COP2 proteins which have been there. So let us assume we are having the ER lumen over here. Okay, the proteins which have been made. Okay, so the resident ER proteins and some of the chaperones which have been there, which are bound to the misfolded protein. So what exactly misfolded protein are? The proteins which are not in the actual shape, and due to which the activity of that particular protein is not well enough. Okay, so for that, the chaperones will be binding to that proteins, and it will fold that particular protein, due to which the protein in the folded state will be activated further once it has been vesicled in the other organelles. Okay, so this ER protein which has been there, there is an exit signal which is there on the soluble cargo protein. So this exit signal, it has been totally been identified by the exit signal cargo receptor. So that is been identified, and overall that proteins are being made. Okay, so that proteins have been packed, and that proteins which have been there, once it has been packed, they are being transported from the ER lumen to the Golgi part. Okay, so this forms are nothing but it is forming a transport vesicle. Okay, and this transport vesicle, it has your SAR1 GTP, it is also having the COP1 coat and so on, and the cargo receptor sequence which is identifying the exit signal. So there may be some of the proteins where the exit signal won't be there, but due to some of the other mechanism, it will be packed and it will be passed on to the Golgi apparatus. So that protein which has been there, which has been packed to the Golgi apparatus, that can come back to the ER through a retrieval system. Okay. So as I said, the coat proteins or the COP2 proteins having the coat, okay, they pass this into the forward direction. Now this is being totally been driven by some of the identical proteins कि भाई ये protein ये जो protein अभी बना है उसको we have to transport from endoplasmic reticulum to the Golgi part. So that is being totally been identified by the V snares and T snares proteins. Okay, now these are the crucial proteins which are there in the COP2 okay vesicle, which identifies कि भाई ये proteins को ही हमको ले जाना है if the signal is found to be appropriate and if there is some identification signal is there in the protein. So that V snares and T snares matching is one of the important component whenever we talk about COP2 vesicle trafficking. As I said, if some of the misfolded proteins are there, if some of the proteins not having sequence or some ER resident protein, so this three condition ho sakti hai. So that is being passed into the retrieval system or the retrograde system where the protein which has been there, that is being identified by the COP1 vesicles and a COP1 coat will be formed. Eventually the retrieval path will take the protein into the back to the ER membrane. Okay, so this is being totally been identified by a sequence called as KDEL. Okay, so this KDEL, it stands for again the amino acid residues. Okay, and this 4 amino acid, it tells us that कि ये प्रोटीन को वापस हमको ER में ले जाना है, चाहे वो ER का रेसिडेंट प्रोटीन हो, चाहे वो मिसफोल्डेड प्रोटीन हो, so उसको हमको वापस ER में ले जाना है. So that is being driven with the help of your COP1 vesicles. Okay, so once this has been there, the Golgi has 3 parts: cis, medial, and the trans part. So from ER it will pass to the cis part. Cis से medial में जाएगा, medial to trans part में जाएगा, and from trans part it will be segregated to the different organelles. Either cell membrane पे जाना तो वहाँ पे भेजेंगे, peroxisome में जाना है, lysosome and so on. Okay. So as I said, in ER we had N-link glycosylation part. In case of Golgi, we are having the O-link glycosylation part. In this, the mannose, glucose, galactose, and N-acetyl uramic acid is being added. Okay. Now this has been done with the help of the enzymes, certain enzymes. For example, if there is addition of mannose, so mannosidase, so the excessive mannosidase, mannose residue बहुत सारा है, so that will be cleaved off with the help of mannosidase, and जो only stabilizing mannosidase होगा, that will be added. Further, N-acetyl glucosamine transferase, that will transfer the N-acetyl glucosamine to that particular part. Okay. Then Golgi mannosidase 2, again it will cleave off and the glucose N-acetyl glucosamine will be added. Overall, what will happen? That function will be there, which will be done by UDP and CMP, which brings down the N-acetyl uramic acid, galactose, and so on. So that will eventually add the residue to the part, to the growing chain. So in this case, already the N-link part which has been done in the ER, यहाँ पे further O-link part होता है. Now this is only for the glycoproteins which have been there, which needs to be transported. Okay, so जो भी ER से signal आई, that has been found, that has been going in the cis Golgi. Okay, and then phosphorylation of oligosaccharides as well as isomer protein occurs. From cis, it goes to cis cisternae part, removal of the mannose residue with the help of mannosidase. Again, it goes to the medial part where the removal of the mannosidase and addition of N-acetyl glucosamine will be there. To the trans part where the galactose and N-acetyl uramic acid will be added. Okay, and finally, in the trans part itself, the sulfation of tyrosine residue and the carbohydrate will occur. from which the protein will be targeted and will be sorted out to lysosome or plasma membrane or the secretory vesicles. So it's a very simple question. Which type of glycosylation takes place in the Golgi apparatus? I think so. This was in 2015, where T-link glycosylation, N-link glycosylation, O-link, and G-link glycosylation was found. I hope you guys know the answer.
O-link.
Yeah. Second question, which of the following is not an example of transmembrane transport between different subcellular compartment? That is transport from cytoplasm into the lumen of ER, transport from ER to Golgi complex, the transport from stroma into thylakoid space, transport from mitochondria intermembrane space into the matrix. Yes. We have been asked
Sir,
transmembrane.
stroma to thylakoid.
Okay. Any other answer?
Yes, sir. Third.
Third is the answer. Why do you think third is the answer?
Because stroma and thylakoid are found in one organelle only. Thylakoid are placed.
Thylakoid are?
Thylakoid are the structures which are placed floating in the stroma itself. So there is no. Oh, okay. That's not the answer.
It is not an example of transmembrane transport between different subcellular compartments.
Yes,
Yeah.
that's from the same compartment.
Yeah, exactly. So it's the same compartment, that's why that answer won't come. Okay, so likewise, this type of confusion options will be there. And whenever we study lot of things, that will be merging with all. Okay, cell biology भी पढ़ा है, molecular biology भी पढ़ा है, likewise. So in that case, we have to think in a more specific manner and more targeted manner. Okay, and then do not hurry to answer. Okay, if the questions are left, it is better that negative marking में हमारे marks ना जाए और इसमें ज़्यादा हम लोग cover कर पाए. So that is the main crux. That is, you have to be more precise कि कौन से में क्या आएगा. Okay, right. Fluorescent tag proteins was used to study protein secretion in yeast. Fluorescence was observed in the Golgi secretory vesicles, the rough ER. Which of the following describe? Sorry?
A option.
A option. Okay. Fluorescence was observed in the Golgi secretory vesicles, the rough endoplasmic reticulum. Sequence of events पूछा है, my dear.
B option.
Sir, B. Second.
Sir, B.
It's second option.
Yeah, so it is जिसने भी second बोला है, they are right. That is basically, it is a sequence of events which has been asked. That is, it is passing from rough endoplasmic reticulum. The secretory vesicles are going to the cis portion of the Golgi apparatus. Okay. Right. So just to have a quick sequence कि कौन सी sequence क्या होगी तो कहाँ पे जाएगा. So ये sequence आपको याद रखनी है. Okay, because ये सब conserved residues है. Okay, so if the protein sequence is having this many amino acids, that is 3, 3, 6 or 8 amino acids वाली sequence है, तो it is imported in the nucleus where the maximum is being driven with the help of proline and lysine. Exported from the nucleus, that is, it is having leucine, alanine, leucine rich region. Okay, imported in the endoplasmic reticulum. Okay, that is having a much bigger sequence जो हमने पूरा sequence signal recognition particle जो identify करता है, and it will cleave off the signal. Exported into the endoplasmic reticulum, that is again lysine, asparagine, glutamine, leucine. Okay, this is a 4 amino acid sequence. Imported into mitochondria, Golgi से जो mitochondria में import हो रहा है, that is overall a bigger sequence. Okay, इसमें majorly the leucine, arginine, alanine, threonine, they are one of the conserved residues. Okay, likewise if it is going to the peroxisome, so it is there are two sequences, PTS1 and PTS2. So PTS2 is the N-link form, whereas PTS1 is more reliable and more it is being researched, where it shows that a C-terminal serine, lysine, leucine rich region has been there. And that is the target sequence where it directly goes to the peroxisome. So on the basis of that, naturally the question also comes, that is the tetrapeptide KDEL is well known as a retrieval signal of several newly synthesized proteins. This process is mediated through specific KDEL interactions. Any single amino acid change in this tetrapeptide is not allowed in terms of its binding with its receptor and its subsequent retention in specific organelle, whereas secretory proteins are devoid of such tetrapeptide. From this observation, indicate the localization of the receptor of this tetrapeptide.
Endoplasmic reticulum.
Yeah, it is the endoplasmic reticulum which has been there. So endoplasmic reticulum, we saw the COP1 vesicles which takes the protein with the help of identification of KDEL. So that KDEL, which is the protein which has been there, that is taken inside into the endoplasmic reticulum. So this KDEL receptor is present specifically in the endoplasmic reticulum, and due to which the protein is taken inside. Okay, so coming on to the next organelle, that is ribosome. Okay, now this ribosomes, we know we are having different in prokaryotes as well as in eukaryotes. If we see prokaryotes, we are having ribosomes are having two subunits, a larger subunit and a smaller subunit. So 50S larger subunit and 30S larger smaller subunit is been there, which is making the 70S complete ribosome in the prokaryotes. So a total of around, you can say 25 lakh molecular weight is the total molecular weight of 70S. As we know, it stands for Sweetberg unit. So with the help of your density gradient centrifugation, the subunits can be eventually being separated, and the overall molecular weight can be determined with the help of that. So once you separate that in the 70, your density gradient centrifugation, you'll be getting either a combined one. Okay, if it is separated 50S or 30S, larger आपको subunit देखने को मिलेगा. So in 50S we are having your 5S rRNA, 23S rRNA, okay, and 34 proteins, whereas in 30S small subunit we are having 16S rRNA and 21 proteins. So this 16S rRNA, it is one of the molecular marker which has been used in various taxonomic and phylogenetic study. So that is also one of the questions जो ज़्यादातर पूछा जाता है कि from 5 kingdom to 3 domain classification, when it is coming, which sequence we are taking for identification of various organisms. So this is the 16S rRNA is one of the sequence which is conserved and which is having very less rate of mutation, and due to which the this is considered to be the marker for phylogenetic analysis. Likewise, for eukaryotic ribosome, we are having the 80S, okay, as the whole subunit, okay, as a whole ribosome that can be dissociated into 68 subunit and the 48 subunit. So 68 subunit, we are having the 3 rRNAs, that is 5S rRNA, 28S and 5.8S rRNA, okay, having the different nucleotide composition. For example, 5.8 is 160, 28S is 4700, and 5S is 120 nucleotide. And we are having around 49 proteins, okay. So a smaller unit, that is 40S ribosomal subunit, that is comprising of only a single one, that is 18S rRNA. It has been presumed and it has been also experimentally proved that this particular sequence of 16S RNA, it is being converted to the 18S RNA form, and it is having the addition of more nucleotide, due to which the molecular weight of this particular increase. So around 1900 nucleotides was being found over here, and it is having your 33 proteins which are being listed. Okay, so if you want to stain this ribosome, brilliant crystal blue is the stain which has been used to study the ribosome organ. We know the ribosomes are been governing the function of protein synthesis, okay, and this protein synthesis is being governed with the help of the overall subunit function. Okay, so yes, we are having the mRNA which has been there, okay, the smaller subunit and the bigger subunit. So this smaller and the bigger subunit, it makes the three cavities which is known as EPA. A stands for amino acid subunit, where the amino acid comes, okay, the peptidal transferase activity, that is stands for P, and E, that is nothing but the exit part through which the tRNA gets exit. Okay. So what happens is, that is your EPA side, in that the tRNA comes to the A side, okay, with an amino acid. So it is called as your charged tRNA. So once the charged tRNA is there, the prior tRNA which has been there, that is forming the polypeptide, that has the polypeptide, it will transfer the polypeptide with the help of peptidase activity. It will transfer to the first one, and that eventually you can see over here 1, 2, 3, 4, that has been formed in the fourth part. So that will again it will shift from A to P side, and this A to P side overall, what is happening is that the A side will be vacated and a new charged tRNA will come. So once this new charge will be coming, simultaneously a 3 will be driven off. Okay, it will be ejected out and it will be emptied. Okay, and that process goes on. So likewise, it starts from your first amino acid, that is AUG codon, which has been identified in the mRNA, and that AUG codon which it brings your methionine. Okay, stop codon, we know that 3 stop codons are there. So stop codon, the polypeptide synthesis will stop, and eventually the dissociation of mRNA with ribosome happens, and finally the new translation starts. So it's a very complex process. In we are not going that much detail in the molecular biology, where the initiation factors are being there. Okay, you will definitely study in the molecular biology part key initiation factors, then termination factors, and so on. That is different for prokaryotes and eukaryotes. So उस पे भी बहुत सारे questions आते हैं. So with respect to this protein synthesis, why we have focused on this? Because there are certain inhibitors which blocks the EPA side. Okay, and overall on the basis of that, the question they are being framed. So if we say that is we are having acting on bacteria, so tetracyclines are the group of antibiotics which are blocking the amino acid tRNA in the A side. Okay, so A side pe block ho jayega, so it is not passed to the P side. Likewise, streptomycin, which prevents the translation initiation to chain elongation, and it leads to the miscoding. Chloramphenicol, it blocks the peptidal transferase action in the P to A. Okay, erythromycin, which binds the exit channel of the ribosome, and eventually it inhibits the elongation of the peptide chain. Likewise, rifamycin, which is there, that blocks the RNA chain by binding to the RNA polymerase. Okay, that eventually prevents the RNA synthesis. If there are certain antibiotics which are acting both on bacteria as well as on eukaryotes, like puromycin, it is causing the premature release of the nascent polypeptide. So complete polypeptide, it is not formed, that is till the stop codon tak nahi form hua, premature release ho gaya. Actinomycin, which has been there, it binds to the DNA and blocks the movement of DNA polymerase, which has been there, that is, it prevents the RNA synthesis. There are certain antibiotics which are acting only on eukaryotes, but they are not acting on bacteria, okay, or other prokaryotes. For example, cyclohexamide, it blocks translocation reaction on the ribosomes. Okay, anisomycin, which blocks the peptidal transferase of the ribosome, and amanitin, that is, it blocks the mRNA synthesis by binding to the RNA polymerase. Okay, so coming to the next organelle, that is nucleus. We know nucleus, it's one of the major organ found in the cell, as the genetic material it is found in the nucleus. So apart from the genetic material, it has the other components also. For example, it has the nuclear membrane. Likewise, just as cell membrane hota hai, plasma membrane. Likewise, this nucleus also has its own membrane. So it's a membrane-bound organelle again. So 2 membranes are present. It prevents permeability. Okay, generally osmotic gradient maintain karta rehta hai. Around 1000 nuclear porins are being found. Okay, so these porins are nothing but pore. It forms the pore through which the mRNA and proteins are being it can go out. Okay, likewise, nuclear lamina or the lamins which are being found. Okay, they are the small. We'll see in detail what exactly it is. It is the intermediate filament, and if some mutation is there, it leads to the muscular dystrophy type of disease. Okay. Then likewise, cytoplasm in nucleus, we are having the nucleoplasm. So nuclear localization sequence which has been there, that eventually it is driving the proteins into that. Karyoplasm, usko nucleoplasm bhi bolte hain, dissolved salts, enzymes and organic molecule. We are having the nucleolus. It is a dense membrane structure, and it contains both the RNA and proteins, and very importantly, it synthesizes the ribosomes. Okay. The last and foremost important part, that is chromosomes. We know that it is having DNA and histones. Okay, DNA which has been wrapped to histones, and the assembly has been there. And the two portions of DNA which has been there, that are the heterochromatin and the euchromatin portion. Simplest question, 2-mark question. I think so. It was asked in 2015. Yeah, the function of nucleolus is, we know that it is for
D.
Ribosomal subunit synthesis. So nucleus, as I said, it is having the nuclear lamina, that is a filamentous structure which has been there, and it provides a mechanical support. So around 10 nanometer in diameter, and it is composed of polypeptides called nuclear lamina. Okay, that is intermediate filaments around 10 nanometer in diameter. And जैसे microtubules, microfilaments में continuous phosphorylation, dephosphorylation होता है. Okay, likewise microtubules में जैसे assembly और disassembly होती है. Likewise in this case also, that is intermediate filaments में also we are having the dephosphorylation, where this basically occurs during the mitosis. Okay, and then the nuclear membrane, it gets disintegrated, and prophase, meta, and telophase में आपको पूरा ये segregation दिखता है. So overall, the mutation leads to the muscular dystrophy, and again, this is a disease where there are numerous SNPs which have been found in case of this nuclear lamina, and due to which the muscular dystrophy is being accounted. So in the diagram, you can see the lamina portion which has been there, okay, and that has been attached to a stronger portion to the part. So lamin A, C, and B, the network of this has been there. And in between the other laminas have also been there, which provides the rigidity to the nuclear membrane. Then we are having the nuclear pore complex, that is a gateway for RNA and proteins. Okay, it contains, it forms a nuclear pore bigger complex, due to which, that is around, you can say that it can easily pass the mRNA and other proteins of larger size. So 15 to 13 times it is the mass of the ribosome, and न्यूक्लियर पोरिन्स जो होते हैं, दैट इज अराउंड 30 डिफरेंट टाइप्स अभी तक हमको ह्यूमन में देखने को मिले हैं. ओके, सो इफ यू सी द स्ट्रक्चर, वी आर हैविंग द आउटर एंड द इनर मेम्ब्रेन. ओके, वी आर हैविंग द साइटोप्लाज्मिक फिलामेंट्स and the intercellular filaments. So this overall, with the help of central framework, it forms a pore-like structure, and the distal ring over here plays a major role. So जैसे polypeptide या mRNA जैसे भी होता है, that is being identified, and it can be easily been passed from one part to the other part of the cell. Yes, mark the incorrect statement about nuclear lamina. Filaments present in the inner membrane of the nucleus, made up of lamin proteins, provide mechanical support to the nucleus. It has bounded ribosomes. Yes? Come on, simple one.
Sir, A.
D.
Yeah, why do you think A?
Sir, I've read somewhere, that's why.
सर साइटोप्लाज्म, सर देयर आर, सर ई कैसे हो सकता है? साइटोप्लाज्मिक फिलामेंट होते हैं, वो साइटोप्लाज्म में होते हैं, लेकिन न्यूक्लियस के अंदर भी तो सर डिस्टल फिलामेंट होते हैं ना.
या इसलिए मैं उसको पूछ रहा हूँ कि ए क्यों लग रहा है. So what is D? किसने बताया हमको? Who said D? Come on. Generally, this lamina, it is not lamins. They are present inside the nuclear membrane. Okay, so they are not bounded with the ribosomes. So that is the incorrect answer. Okay, so a glimpse of the molecular biology, that is the structure of DNA, just going superficially about that. As on the basis of this, again the questions are there. Okay, so we know there are 3 forms of DNA which has been found, A, B, and Z form, or the Z form. Okay, on the basis of some of the attributes which have been there, they are being classified. For example, the type of helix which has been there, in case of A and B, it is right-handed helix, whereas in case of Z form or Z form, it is left-handed. The repeating unit, if you see, it is in the A form, 1 base pair is been one of the repeating unit, whereas in Z form, it is 2 base pair. Rotation, if you see the overall rotation, that is 32.7 degree of rotation per base is been found. In case of B, 34.3, and in case of Z, it is 60 degree. Whereas mean base pair per turn, that is around 11 residues. Okay, likewise in B, it is 10.5, and Z, it is 12 residues. Okay, this is one of the commonest question which has been asked in 2-mark question also. Inclination of base pair to axis around 19 degree. It is around minus 1.2 degree, whereas in Z form it is minus 9 degree. Okay, likewise glycoside angle is nothing but the angle at which your phosphate and sugar is been arranged. So if the phosphate is arranged in the reverse direction, it is called as anti-direction. If it is arranged in the similar direction of your sugar, it is called as the same direction. So in case of A and B form, both are anti. So glycoside angle is anti. In Z form, it is the purines are syn and pyrimidids are anti form. Nucleotide phosphate to phosphate distance, if you see, so it is around 5.9 Armstrong, whereas in case of B form, it is slightly separated, 7 Armstrong. In Z form, it is 5.7 for C and 6.1 for G. Sugar puckering, that is more inclined towards which carbon? Okay, phosphate group. So in case of A form, it is the endo form. So C3 में endo देखने को मिलता है. Okay, in case of B form, it is C2 endo group, whereas in Z form, it is C2 endo for C and C3 endo for G. Okay, and finally the helical diameter, that has also been asked, 23 Armstrong or 2.3 nanometer in case of A, 20 Armstrong or 2 nanometer in case of B, and Z form 18 Armstrong or 1.8 nanometer in case of Z form. So one of the commonest question 2019 में पूछा था, it was a 4-mark question. A form and Z form of double-stranded DNA differ in the handedness of their helix, nucleotide sequence, and configuration of the base to sugar. Based on these properties, which one of the following statements define a correct combination for A and Z form of DNA? Okay, so 2 minutes, just quickly go through the option. The first one says right-handed double helix and anti-configuration for the base to sugar arrangement in aDNA, and left-handed double helix with alternating sequence of G and C as a general pattern, and alternating syn and anti-configuration for the base to sugar arrangement in the Z form. Right-handed double helix and syn configuration for the base to sugar arrangement in aDNA, and left-handed double helix with alternating A and G sequence and anti-configuration for the base to sugar arrangement in the Z form.
Sir,
Left-handed. Yes, well.
the correct answer is 1.
Why?
Because there is right-handed in the case of A and B configuration of DNA, and it is left-handed in the case of Z. And in the last slide, we only observed the 2 letters G and C in case of the alternating sequence. So it goes with first correct option.
Yeah, what's your name, my dear?
Dhimahi.
Dhimahi.
Yes, sir.
Yeah, Dhimahi, I think so. You are right. And that is the correct answer with respect to the A and Z form. Okay, likewise, B and Z form के भी questions है. Okay, with respect to that, what is the Armstrong, the base pair turn, वो दो mark के questions उस पे से आते हैं. Okay, so coming on to the import and export through nuclear pore. Okay, whenever we say import and export, as we saw in endoplasmic reticulum, then we saw in mitochondria, then from Golgi and so on. Now, this import and export is also governed in the nucleus for the nuclear proteins. Okay, so this import and export are being governed with the help of the proteins called as your importins and exportins. So this importins, they bind to a GTPase called as RAN-BP2 GTPase. So once this has been bound, okay, the nuclear pore which has been there, it identifies the RAN-BP2 and your importin alpha and beta. Together with the help of that, the pore will allow that particular cargo to go inside. Okay, once it is going inside, again the hydrolysis of GTP happens. So RAN GTP to RAN GTP happens. That is again the GTP, your phosphate is being cleaved and your RAN is getting inactivated. So once the cargo is being there, it is doing its own function in the nucleoplasm. But let us assume some of the cargo which the nucleus has to transport outside. Okay, so that cargoes are again identified by the exporting proteins and other importing proteins, basically the re-exporters which have been there. So this re-exporters which are there, it combines with the exportin and the cargo molecule, okay, with the help of your importin beta. So it goes outside, again the nuclear pore will identify that, and this nuclear pore, once identified, the cargo will go in the cytoplasm, the cargo will get separate. So wahan pe the role is played by your RAN gap, where RAN gap, again it is a GTP which is hydrolyzing the GTP, and the RAN GDP is being produced. So in short, there are some of the proteins which are playing a major role, like as in case of mitochondria, it was Tim and Tom proteins. In case of this, it is your RAN GTPase and the RAN gap proteins. So RAN over here, it is activated, yes, once it is GTP bound, and it is inactivated when it is GDP. It is converted to GDP. So more the GTP, more the RAN will be activated, less it won't be activated. So GDP hydrolysis हो जाती है, तो GDP में convert होने के बाद the RAN will be in active form. Again, the question for A and B form. Consider the following statements. A form of the DNA has 10 base pair per turn, and A form of DNA has 2.3 Armstrong helix rise per base pair. Both the A and B form of the DNA have wider major groove and narrow minor groove. The crystalline nature of cellulose 1 about alpha 1,4 linkage between the glucose subunit. The double bond in the anterior lipids are always cis, which provides fluidity of the plasma membrane. Which one of the following combinations are correct? So, in short, what they have done, that is they have combined all the things. That is, they have combined some of the molecular biology stuff, some of the biochemistry stuff, as well as the cell biology stuff. Yes, Swapnil, why do you think it is C?
Sir, because C option is incorrect, rest I don't know, but the answer to this question in third option only C was not there. Rest in 3 options C is there, but it is an incorrect answer. So according to me, the answer should be third option.
A and D.
Yes, sir.
And what about?
Because in cellulose, beta linkages are present instead of alpha linkages.
Right, exactly. So that is the wrong answer. And why don't, मतलब B आपने क्यों सोचा नहीं? B statement.
Sir, I was only sure about only the option C. The rest all options I was not sure. But according to the options, only C was not present in the third. So
Okay.
I guess.
Okay. Generally, A and B form, they both have the similar major and similar minor groove. Okay, so ऐसा कुछ नहीं है. Yes, the major and minor groove will differ with respect to Z form. Okay. The next is the lysosomes, the suicidal bags, which have been there, one of the important organelles in the cell. Okay, it is responsible for digestion of all the macromolecules, the old cell parts which have been there, as well as the small microorganisms. So they are being surrounded again by the membrane, and that membrane maintains the acidic environment. So that is a characteristic of this lysosome. Majority it is having the acidic environment. So that is being driven with the help of your proton pump. Okay, it is carrying various hydrolytic enzymes, that is acid hydrolysis, macromolecules like nucleic acid, protein, and polysaccharide. So even questions like on mutations on acid hydrolysis, they may come. Okay, likewise, थोड़े mutations अगर ऐसे हो रहे हैं कि उसके वजह से what are the different storage diseases which are being found. Okay, so if you see storage disease like Gaucher and Niemann's pig disease, which is due to the sphingolipids, okay, accumulation, which is happening. Likewise, Tack-Shack disease, which is gangliosidosis and lipids, they both are accumulated in the cell. Hunter syndrome and Hurler's disease, that is glycosaminoglycans, which are being accumulated inside the cell. So if you see, this lysosome plays a major role in driving off the debris, means the cleaving of the unwanted part, okay, and digesting the unwanted plant. So we are having, for example, we are having an antigen or a bacterium which has been taken inside, okay, with the help of receptor. So phagocytosis has been done by the cell, let us assume a macrophage or a neutrophil cell has been there. So the lysosome of that cell, it goes and it forms a phagolysosome with the help of in the bacteria. Okay, and phagolysosome has been formed. So this phagolysosome, it will the lysosome in that it will release its acidic enzymes, so proteases and so on. So that will be released and it will clear off the bacteria and the debris will be exocytosed. Okay, so on the basis of that, we call that particular procedure as a autophagy procedure. That is the function of a lysosome. So 4 types of autophagy which are being found for this. So one is called as a macroautophagy. Macroautophagy, it is nothing but the lysosome which is being there, okay, that is engulfing the bigger part. उससे double size का वो bigger part engulf कर रहा है. Okay, so in this phase, what is happening? Your mTOR-C1, that is one of the protein which is attached to lysosome, and it is stopping the macroautophagy. Okay, but yes, there is when a nutrient depletion or nutrient replete condition will be there. Some of the condition will change, and this mTOR-C1 will dissociate from the lysosome. So what will happen? That will initiate the fusion or the fusion of this lysosome with the molecule. Okay, so that will eventually be driven with the help of the proteins, as we discussed in case of COP2 vesicles, that is snares protein, that is VMT snares. Likewise, in this case also the fusion has been governed by the snares protein. Some of the tether proteins and some of the protein like LC3 and RAP7, which fuses the lysosome and your autophagosome. So once it has been fused, there is degradation, and degradation starts with the release of the enzymes. Okay, and that has been done. So after that, the autophagic lysosome reformation occurs, where the clathrine plays a major role. Okay, and that has been overall cleaved. The microtubule over here, which has been formed, that has been cleaved with the help of a DYNE2 protein. Okay, and KIF5B protein, which has been there. So this protein, it is doing the phosphorylation on this, and that will eventually activate DYNE2, which is a cleavage protease, and it will cleave the microtubule assembly in this case, and hence the lysosome will be reformed. So that is your macroautophagy. Just remember, macroautophagy कब होता है? जब bigger size वाले molecule आते हैं. In case of chaperone mediated autophagy, there is a sequence called as KFERQ motif, which is being identified by your LAMP2A receptors. Now this particular receptor, it is forming a translocon. So how it can be done? That is with the help of your HSC70 and HSP90. So chaperone binding to this and to identifying that particular motif, eventually that motif will be gone inside the lysosomal lumen, and then it will be translocated, and then it will be cleaved off. So these are, you can say, these are for the small macromolecules which have been there. So that is being driven with the help of chaperone, that is HSP90 and HSC70 and so on. Okay, and again the translocon disassembly has been there, so that excessive other molecules are not taken inside for degradation. Then we are having RN or DN autophagy, where majorly the nucleic acid molecules have been degraded. So again, that is being driven by LAMP2C, which passes the nucleic acid to your CYT2. So this CYT2 is specifically for nucleic acid, and it will form the translocon, and that will go inside. Okay. Then next is we are having the microautophagy. Now microautophagy is simpler one, that is your microsome formation. And this microsome formation which has been there, that has been nothing but the invagination of the membrane, and eventually the molecule is going inside. Okay, so for this, this is the simplest example which has been found in the microsome system, where the HSC70 and it identifies the phosphatidyl serine, which is being labeled as PS over here. So that PS has been identified and it has been taken inside. The whole lysosome has been fused, and finally it has been degraded. Yeah, so coming on to the question, in Taxa's disease, accumulation of glycolipids occurs especially in the nerve cells. These cells are greatly enlarged with swollen lipid-filled endosomes, and the children with this disease die at early age. Such conditions occur due to the defect in, again, I said Taxa disease is a storage disease. So with respect to that, yes?
A. Option A.
Yes, why do you think it is A?
So because it is like being caused because of the storage issue of the gangliocytes that is there in
exactly. Yeah, exactly. Right, the next question, a null mutation is created in a gene which is responsible for specific phosphorylation or at 6th carbon position of mannose on acid hydrolase occurring in the cis Golgi. The following statements are given towards explaining the effect of its mutation. The lysosome will be devoid of lysosomal enzyme. The lysosome enzymes will be secreted out. The lysosomal enzymes will get localized in the cytoplasm. Which statement or combination of statement will explain the effect of mutation if the acid hydrolysis in the mutant do not get degraded? So there are two conditions over here. There is a mutation happening in acid hydrolysis, which is found in cis Golgi. So cis Golgi से medial से trans पे pass हुआ, trans से फिर वो lysosomal में pass होगा. Okay, that is the condition. So lysosome में आके फिर क्या होगा? Whether the lysosome won't be having that particular enzyme, or it will be secreted out, or it will get localized in the cytoplasm. Mannose residue पे है. Yes. Come on. Any hint? Any wild guess?
A and C, the option A.
Option A. Why?
Sir, because of null mutation, the protein will not transfer to the lysosomes. So lysosome will be devoid of the lysosomal enzymes, and that enzymes will be localized in cytoplasm.
जनरली क्या होता है अगर मैनोज रेसिड्यू में कुछ ऐसा पोजीशन पे मतलब फॉस्फोराइलेशन मींस if the mutation is happening, okay, naturally lysosome won't be having that particular enzyme, okay, and if at all it passes into lysosome, it will not identify its own enzyme and it will be secreted out. Okay, so secreted out होके localize मतलब localize aggregate होगा cytoplasm में, but it will not localize previously then cytoplasm. पहले वो lysosome में तो जाएगा ही जाएगा, and after that it will secrete out. Okay. Chalo, coming on to the last few two organelles, that is peroxisome and plastids. Okay, coming on to the peroxisome, it's a semi-permeable membrane, urate oxidase crystal core, and it uses molecular oxygen basically to remove the hydrogen atoms which have been there. Okay, and so वहाँ पे highly oxidized state होता है, oxidation reactions are maximum. Okay, and this particular organelle produces hydrogen peroxide, that is oxidative enzymes drive this particular reaction. So catalysis, for example, it uses this H2O2 and it forms the nothing but your more soluble form, water. Okay. Peroxisomes, they have been found in plants where they are active at photorespiration. They are found in the, they are called as glyoxysomes in again plants. They do undergo, they have a cycle called as Glyoxylate cycle. So glyoxylate is the metabolite which has been produced over there. Lipid biosynthesis has been driven in this. Seed germination, that is it stores fatty acids and carbohydrates. Okay, it also converts majorly the the higher form, that is polysaccharides and so on, and degradation of purines, that is catabolism of purines is happening in this case. So DAB, that is 3,3-diamino benzazidine staining is being done to understand peroxisome. A 2-mark question: which one of the following statements is not true? Beta oxidation of long chain fatty acid occurs in mitochondria. Fatty acid biosynthesis occurs in peroxisomes. Peroxisome utilizes H2O2 to oxidize a variety of substrates. Peroxisomes import their repertoire of protein using sorting signals.
So it should be A.
The option B.
A.
Option B.
क्यों ये लग रहा है? Yeah, it is second option. Biosynthesis do not occur in peroxisome. Where does it occur?
In cytosol.
Sure. Come on.
Mitochondria.
Mitochondria. We saw. Okay, so coming on to the last part of the presentation, plastids. Plastids are, though there are very, very few questions on this. So plastids are of many types. For example, etioplast. They are nothing but the prolamellar bodies. Okay, that is, they are precursor of chlorophyll. Chromoplast, red pigments maximum होता है उसमें. Okay, reddish to orange pigment. This pigment synthesized are been stored in this case. Chloroplast having chlorophyll. Okay, we saw chloroplast much in detail. They do photosynthesis and lipid biosynthesis. Leucoplast, synthesis of fatty acids and amino acids. Amyloplast, storage of starch and sugar. Oleoplast, storage of lipids and oils. And proteinoplast, that is sites for enzyme activity, that is majorly the proteins. Okay. So till now, till 2020, what I saw was the majority of the questions, they were diverted from, they are from mitochondria, endoplasmic reticulum, lysosome and Golgi. Though अभी trend ऐसा आ रहा है from past 3 years, nucleus पे ज़्यादातर questions आने लगे हैं, nucleus and chloroplast, and others बाकी जो हमने अभी तक पढ़ा उस पे थोड़ा कम है. This is only for my topic and my analysis. You can have your analysis also. But 2013 से या 12 से अभी तक जो questions आ रहे हैं, that is I have made a tentative pie chart from the analysis. Okay, so इसका मतलब ये नहीं है कि हमको टॉपिक छोड़ देना है. We have to read the topic. Okay, but yes, you can have a more focus accordingly. Okay, thank you so much. On this slide, my email ID and my number is there. So for any query, you can ask me. Okay, I hope you guys must have enjoyed the lecture. Though in 2 hours, I need to complete all the organelles and all the parts, or it is one of the toughest job. But I have concentrated mainly on the CSR part, means CSR में कैसे questions आते हैं and so on. So वो question पे concentrate करके ये information दिया था. Yes, I hope any question. If any question, I can solve.
Any questions, students?
No questions from our side.
Okay, so I think I'll take this opportunity to thank Dr. Parth for his very detailed presentation. And the good part is that, through this 2-hour session, you have got a glimpse into all the important aspects associated with cell organelles. And that is the main motive of this crash course workshop, that it helps you to get into the details, otherwise it becomes a concern. So I'm sure you can, private your queries if any, even after the class. I'll be sharing the details of the presentation also with you. And apart from that, we will also share some practice questions. So you may all follow it up. And still, if any doubt persists, then I'm sure Dr. Parth Pandya will be happy to solve them. Okay?
Definitely.
Thank you, Dr. Parth. And I'll close the session here. I'll stop the recording first. And we will keep you updated with the next set of topics. And